can't walk to the corner store or local pharmacy and pick up a bottle of medicine that cures blindness or fixes sickle cell syndrome or prevents Parkinson's. Yet. Those miracle cures are not as far off as you might think, thanks to a recent discovery known as CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is a way of describing the orientation of the DNA base pairs in chromosomes. CRISPR in of itself is actually a defense system bacteria use against invading bacteriophages. If a bacterium survives a bacteriophage attack, it will activate its CRISPR system and store a section of the phage RNA in a genetic databank. When the same type of phage attacks again, Cas9, a protein machine used in the CRISPR system, attaches to a copy of the stored phage RNA. At this point, Cas9 will skitter across the bacteria's DNA, trying to find an exact replica of the RNA it's attached to. When it does, it snips it off, rendering the phage's gene useless. This is much more effective and precise than any gene editing technique humans have ever used, and we've improved upon it, using a similar protein called Cas12 or CPF1. CPF1 requires less RNA and makes it easier to introduce genes to the split DNA strand. So how does CRISPR relate to a cure for blindness, Parkinson's, and sickle cell syndrome? It involves gene therapy, which is a medical procedure in which healthy cells are placed where unhealthy cells were or are. Eventually, the healthy cells proliferate enough to become the majority. CRISPR gives us a broader spectrum of what kind of healthy cells we could make and what kind of things we could cure. CRISPR was discovered in part thanks to ocean bacteria. Several mappings of the bacteria found in the Alicante regions of Spain was happening when Francisco Mojica, studying Hylophranax R4, happened to find several repeated parts of DNA that were short and palindromic. The reason for this would remain a mystery for many years to come. CRISPR's true potential wouldn't be unlocked until 2012, when it was discovered it could gene edit inside of its environment. However, it truly came to power when Jennifer Doudna and Emile Charpentier found out a way to make it gene edit outside of its natural environment. Finally, we could use it to gene edit plants and animals.
Even now, companies like CRISPR Therapeutics are using a clinical trial to test Cas9 CRISPR gene editing to cure beta thalassemia, a blood disorder. CRISPR Therapeutics is one of the emerging companies to use CRISPR. Among its ranks are Intellia Therapeutics and Adidas Medicine.
All these companies were founded recently, sometime just after 2012 actually. Now if you've been paying attention, you'll know why. It's because CRISPR, as a gene editor, only became viable in 2012. In fact, the company that first decided to use CRISPR was CRISPR Therapeutics.
CRISPR Therapeutics was made in early 2013 by Dr. Roger Novak, Emile Charpentier, and Sean Foy. It's their mission statement to make a gene-based therapy for degenerative human diseases. And they need a lot of researchers for that. In fact, they need researchers both for stem cell and chemical delivery, scientists of analytical developments, quality control directors, clinical coordinators, and many more. Just one job, senior research associate in a stem cell engineering group, requires all this stuff. That's just the minimum.
But would you want to even work in that field? Or in any job involving CRISPR? CRISPR has been the main topic of many ethical dilemmas. CRISPR is very powerful. It has the power to change humanity as a whole. It allows us to decide what we want in a living being, how they look, how they act, and so forth. The beginnings of CRISPR will obviously be about eradicating horrible diseases, but it won't stop there, many ethicists speculate. They believe this technology will be used for godly purposes, to make humans stronger, more beautiful, more intelligent. Ethicists worry that this will allow for even greater divide between the rich and the poor, a genetic divide. Right now, all we can do is take caution and proceed forward with this dazzling new technology, and one can only wonder, what will the future hold?